Well, thank you, Phil, for the lovely invitation. It is indeed a pleasure to have the opportunity to present our work to an international metabolism community. Our lab works in cancer metabolism, and one of the focuses of our lab, one of the cancers that we study, is pancreatic cancer. So today I will be presenting a recently published story from our lab, talking about how pancreatic cancer cells can adapt to extreme nutrient deprivation. Tumor growth is complex, and that's simply because it grows in a host and not in a tissue culture dish. And recent work from our lab and others has highlighted the relevance of studying tumor metabolism in a physiological context, as discrepancies between in vitro and in vivo work are being increasingly underscored and recognized in the metabolism field, and that could impact their translation into the clinic. So in some aspects, I tend to think of a tumor as if it were an organ, could be thought of as an organ. So not only is a tumor affected by its own microenvironment, but also by the systemic metabolism of its host, where it is growing. A host has a defined systemic metabolic state. It could be lean, it could be obese, it could be insulin resistant, diabetic, athletic, etc. It is therefore not unreasonable to think that the host systemic metabolic status can influence the growth and metabolism of the tumor, and vice versa. And indeed, several epidemiological and other correlative studies have provided evidence for these forward and reverse arrows here. For example, obesity is a systemic metabolic state that we know is associated with enhanced incidence and mortality from several cancers, especially the gastrointestinal cancers. For the reverse arrow, one can think of cancer patients in advanced stages of their disease, where they start wasting their muscle and fat, and that is thought to be caused by the mere presence of the tumor in their body. So the tumor here is affecting the systemic metabolic state. So our lab is interested in questions in this field, working on both arrows, forward and reverse. And we have studied obesity and tumor growth and metabolism in the context of pancreatic cancer in the past. And we have several projects at the moment in the lab working on cachexia or body wasting in pancreatic cancer and lung cancers. So why is that relevant to anyone or to people that work on cancer metabolism? Over 20 years ago, Doug Hanahan and Robert Weinberg published a hallmark paper describing and listing the hallmarks of cancer. And those included sustaining proliferative signaling, evading growth suppressors, resisting cell death, induction of angiogenesis, enabling replicative immortality, and invasion and metastasis. However, 10 years later, they updated their hallmarks of cancer, adding 4 new ones. So in addition to genomic instability, inflammation, and evasion from immune destruction, which is the basis for immunotherapy, they also added deregulating cellular energetics in cancer. So in other words, this is altered metabolism or cancer metabolism field. So cancer metabolism or altered metabolism is a bona fide hallmark of cancer, and that's why we do care. The overarching goal of our lab can be represented in a very simple question. Can we harness in vivo metabolic dependencies in tumors for therapeutic benefit? So we focus here in this story, and part of the lab works on pancreatic cancer. Pancreatic ductal adenocarcinoma, or PDAC, is the major form of cancer and is a highly lethal malignancy, as you probably know. 5-year survival rate is about 9 to 10%. It does not respond to therapy. It is diagnosed really late when it has already most of the time metastasized, and the only option for cure is if it had not invaded the lymph nodes and can be resected completely by surgery. And that is a very, very tiny population of the cancer patients that have pancreatic cancer. We're very interested in PDAC because it's really unique. It's a very unique tumor. Its uniqueness stems partly from the fact that the tumor mass, it is estimated that only about 10% of the whole tumor mass is cancer cells, and the rest is stroma. It is very, very densely compacted stroma consisting of fibroblast matrix that is secreted by the cancer cells and the cancer-associated fibroblasts or stellate cells, macrophages, other immune cells, endothelial cells, etc. So I tend to think of it as if this tumor is choked, and this dense stroma is restricting the access of blood vessels and nutrients to the cancer cells. How does PDAC then survive, not only thrive under such harsh nutrient-deprived conditions? And so it has been recognized for a while, that has been published, that PDAC relies on unconventional means for acquiring nutrients and surviving and growing. And two such processes are autophagy, or the recycling of nutrients, intracellular molecules, components, and the second is macropinocytosis, or the uptake of extracellular macromolecules such as proteins, chopping them up and then remaking whatever nutrient it needs to grow. However, we are really not sure whether these two processes are sufficient for PDAC to survive and grow aggressively under these nutrient-deprived conditions. So this was the basis of our project. So we tend to think that under such harsh conditions, nutrient deprivation, the cancer cells are perhaps under high pressure, which leads them to acquire genetic mutations and, or, it's not mutually exclusive, acquire non-genetic adaptations to deal with this nutrient deprivation and adapt to it and grow. And so in this project, we are trying to target adaptive mechanisms that pancreatic cancer cells undertake to overcome nutrient deprivation. A graduate student from the lab, her name is Peyun Tsai, who already left, and a postdoc in the lab in sick leave, teamed up on this project and asked this very simple question. So although multiple studies had looked at the deprivation of pancreatic cancer cells or other cancers of one of their preferred nutrients, either glucose or glutamine, and found out that they would then depend on the other nutrient or other nutrients that are present, we don't know, not to our knowledge, have we seen study that had looked at the deprivation of cancer cells, or particularly PDAC cells, of the two most abundant and preferred tumor nutrients that are glucose and glutamine. So they asked, can and how do PDAC cells survive glucose and glutamine deprivation? So this is how the project started. So at the beginning, the graduate student Pei took 7 PDAC, human PDAC cell lines listed over here, and harshly treated them or cultured them in a customized low glucose, low glutamine medium. The concentrations are listed here: 0.5 millimolar glucose, 0.1 millimolar glutamine. That represent about 20-fold decrease from the levels that are normally found in the standard RPMI culture medium. And to give you an idea, these levels are about 5 to 10-fold lower than the levels of these nutrients in the human serum. Why did we go lower than the human serum? So of course, I stressed on the fact that now it is very important to study cancer metabolism under in a physiological context. So if it's not in vivo, you could perhaps mimic that by lowering the levels that we use in standard tissue culture to those that are present in the human serum. However, these may not accurately reflect the microenvironment of the tumor, because the tumor has much less access to these nutrients. And therefore, and this is evidence in studies that have shown that the tumor cells and the cancer cells in the core of tumors have lower availability of nutrients such as glucose and glutamine as compared to the periphery. And also that cancer cells in the cancer tissue, they have less availability to nutrients than adjacent normal tissues. So this is why we went lower than 5 to 10-fold lower than what is known to be present in the circulating serum in humans. And so after she did that, we looked at what would happen. And as everybody had expected and told us, oh, all of the cells are going to die. And indeed, within 2 weeks from treating the cells with this harsh low medium, low glucose and low glutamine, the majority of the cells died. However, we were very excited to see that rare single cells started to emerge and grew gradually throughout the days and the weeks, and we were replenishing the medium every 2 to 3 days. 2 to 3 of these cell lines predominantly grew clones. And we chose the top 2, SU2 and A9AT. Miyapaka was the third. So Pei looked at these cells that formed clones, and she called these clones adapted clones or A clones. And then to study them, she compared them. So she selected out of these adapted clones 3 clones. So you're going to see that these are called C1, C2, C3, 4, 5, and 6. So as a comparison, as a control, she selected clones of parental cells that were grown under high medium, so high glucose, high glutamine. So she plated them at very low dilution so that they would form, she would let single cells form clones, and then selected these clones in a similar manner as she did for the adapted clones. And she called these the non-adapted or NA clones, 1, 2, 3, 4, 5, and 6. Then we asked the question, is there a differential proliferative capacity in the adapted clones as compared to non-adapted clones? Do they have enhanced fitness in vitro? So we performed several in vitro studies, starting with a 7-day cell proliferation assay without media replenishment. And as you can see, there was an evident increased fitness in the proliferation of adapted clones, which are in red, as compared to non-adapted clones over here. And I'm only showing here one of the sublines, A9AAT cells, but SU2 had a similar phenotype. They also had lower cell death and formed a larger number of colonies in a colony formation assay. And when plated under three-dimensional conditions, they either formed a larger number of spheres with a larger area, like in A9AAT cells, or they formed similar number of spheres, but the adapted clone spheres had larger area. So obviously, the adapted cells had enhanced proliferative fitness in vitro as compared to non-adapted cells. So then we asked, does this improved fitness in vitro translate in vivo? So to address this question, we injected some of these clones orthotopically into the pancreas of immune deficient rag mice. And then we followed them by imaging by ultrasound, and we asked whether there was a difference in the arisal of these of tumors, and whether all injections generated tumors, and whether the tumor size was different. And indeed, what we found was that only 8 out of 13 of the non-adapted clones generated tumors within a span of 6 weeks. And that was with a delayed latency, as you can see, from 3 to 4 to 6 weeks. It took a long time for us to be able to detect these tumors, and they were on the smaller end. Whereas the adapted clones, 14 out of 14 of them, they were derived from 2 different clones, generated tumors that were detectable even at 3 weeks, so it's much shorter latency to detect the tumors by ultrasound. And the tumors were about 4-fold larger in volume and about 2-fold heavier than the non-adapted ones. Moreover, 2 of the mice that were carrying tumors from the adapted clones succumbed to the tumor burden. They died by week 5, so we were not even able to include them in this average. So this average is actually underestimating the size of the tumors that arose from the adapted clones. So far what I've shown you is that adaptation of pancreatic cancer cells to low glucose, low glutamine media endowed them with enhanced proliferative fitness in vitro that also translated in vivo, allowed them to grow much faster. It's like they were primed for growth under nutrient-deprived conditions. So before I go further on the mechanism, this is just to show you that at sacrifice, you can see that the tumors arising from the adaptive clones were at least twice heavier than the ones arising from the non-adaptive clones. And this is a representative image of the pancreas. This is the spleen. This is the liver. This is in a non-adapted clonal cells were injected in this mouse, whereas the adaptive clones, you can see that at sacrifice, the whole pancreas was transformed into a tumor. Now, to investigate, try to identify metabolic pathways or mechanisms that could explain this enhanced fitness acquired by the adaptive clones compared to non-adaptive clones, we performed both RNA-seq, so transcriptomics, and metabolic profiling on the adaptive non-adaptive clones from the SUP2 cell line. And transcriptionally, there was enrichment in several pathways, including purine and pyrimidine, the nucleotide pathways, also amino acid metabolism, and mTORC1 pathway. I'm not showing it here. What I'm showing in this slide over here is metabolic profiling. And as you can see, independent of adaptation under nutrient-replete conditions, so high conditions, all clones clustered together. They were very similar if you look at these pathways: amino acid metabolism, nucleotide biosynthesis, TCA cycle, and glycolysis. However, when we shock them with a low medium, low glucose, low glutamine, if you look at the non-adaptive clones right here, there's a dramatic change, alterations in the metabolism in all four pathways. Right there, amino acid, nucleotide, everything is changing. So what is happening here, you would say, of course, if you deprive the cells of nutrients such as glucose and glutamine, they're going to induce autophagy, and therefore they have much higher levels of all amino acids, except for here alanine, glutamine, glutamate, and aspartate, which is consistent with glutamine deprivation. And yes, that is true. And then what we see over here is an accumulation of mono and dinucleotides and their precursors, indicating or suggesting that perhaps there is lower DNA synthesis. And there's a decrease in the metabolized from the TCA cycle and from glycolysis. If we look now at adapted clones, however, under low conditions, they look so much closer to all the clones that were grown under, they were treated with high conditions. So it's like this phenotype of the non-adapted cells, non-adapted clones under low conditions, the shock, metabolic shock that I would say for these cells is suppressed or mitigated upon adaptation. You can see that there is still indeed higher levels of amino acids in the adapted clones under low conditions, but it's much lower than in the non-adapted clones, suggesting perhaps that these adapted cells perhaps are using these amino acids as a nutrient source to synthesize whatever they need for proliferation. Similarly, the nucleotide biosynthesis profile looks much closer to the one that we see under nutrient-replete conditions right here. And that is consistent with cellular proliferation and DNA synthesis. Again, we are seeing here lower levels of mono and di, and accumulation of dry nucleotide phosphate. Now, again, TCA cycles similarly, and for glycolysis, there's a partial reversal as well. So we focus on these two metabolic pathways, or the metabolism of amino acids and nucleotides. And so to confirm that indeed there was increased DNA synthesis in the adapted more proliferative cells, we confirmed it by feeding the cell C14 aspartate, which integrates into pyrimidine synthesis. And you can see here that there was doubling of the incorporation of C14 from the C14 aspartate into DNA in the adapted clones as compared to the non-adapted clones under low conditions. Because we know that mTORC1 signaling can affect synthesis of pyrimidines through, this is the work of Brendan Manning, through its direct substrate, S6K. When S6K is active and phosphorylated, it can activate and phosphorylate the rate-limiting enzyme for pyrimidine synthesis, that is CAD, and it phosphorylates it on serine 1859. And so we wondered whether mTORC1 was active to explain this phenotype that we're seeing in the adapted cells. And indeed, we were very surprised to see that the adapted clones, as compared to the non-adapted clones, sustained active mTORC1 signaling despite glucose and glutamine deprivation, as you can see over here in the phosphorylation of S6K and phosphorylation of 4-EBP, which are the immediate substrates of mTORC1. And consistently, because we know that CAD is a substrate of S6K, we saw that it was phosphorylated on serine 1859 in the adapted clones, but not in the non-adapted clones. So now we know that these adapted cells have sustained active mTORC1 signaling, and that they have increased pyrimidine synthesis, DNA synthesis, and an alteration in their amino acid metabolism, leading to decreased levels as compared to the non-adapted clones under low conditions. Okay, so again, showing the signaling. Now that we talked about the nucleotides, when I move towards the amino acid that I just mentioned, and like I just said, these levels, even though they were still higher than in, so if you compare, it depends on how many groups you have, you will see that the differences change. So here we're only comparing two groups, the non-adapted, adapted under low, whereas in my previous slide, I was showing 4 groups, so high, low, non-adapted, adapted. So you will see here, as you saw before, there's mitigation or suppression of this increase in amino acids in the adapted cells. And we thought that that would be the result of usage of these amino acids. So now, comparing them next to each other, just as 2 groups, you will appreciate this difference much more. Again, so the adapted cells have lower levels of amino acids under low conditions. We particularly wanted to focus on the leucine, valine, isoleucine, the branched chain amino acids, because we know that branched chain amino acid transaminases could transfer the nitrogen from these to glutamate and to aspartate, and that we knew that aspartate is necessary for the synthesis of both purines and pyrimidines. So we thought that perhaps these cell-adapted clones are using the amino acids, including the branched chain amino acids, for synthesizing, for using the nitrogen for nucleotide synthesis. So the idea was that, for example, if we were to trace a 15N labeled leucine, so heavy isotope labeled branched chain amino acid, through the activity of BCAD, the branched chain amino acid transaminase, it would transfer the nitrogen into glutamate. And then from glutamate, the aspartate transaminase could transfer the nitrogen into aspartate. And aspartate is needed for purine and pyrimidine synthesis. But also glutamate could serve as a substrate for glutamine synthesis if glutamine synthase is active. And you need the condensation of ammonia with glutamate to generate glutamine. So indeed, we found that the nitrogen from leucine, so this is a mass shift M+1 with mass spec, you can see that this nitrogen is transferred into glutamate, indicating that the transaminases are active to generate glutamate, and then from glutamate to aspartate, so through GOT, the aspartate transaminase, and that also we see glutamate synthesis from this glutamate, because the M+1 is also present here, particularly in the adapted clones as compared to the non-adapted clones under low conditions. And this nitrogen can also be traced into metabolites involved in nucleotide synthesis. As you can see over here, a significant or dramatic difference between the adapted and the non-adapted clones. Okay, so now we proved that we had evidence that the amino acids were serving as a source of nitrogen for nucleotide synthesis, and that glutamine was also being synthesized. So we asked, is the main enzyme that is required for synthesis of glutamine induced in these adapted clones? So you can see here that this is a Western blot, that only the adapted clones under low conditions showed significant levels of, so there's a very little detectable level of GS in the non-adapted clones under low-low, but this is much more evident in the adapted clones under low-low conditions. So glutamate dehydrogenase is the first step in glutamate synthesis before glutamine synthesis. It catalyzes the reductive amination of alpha-ketoglutarate to generate glutamate. And then condensation of ammonia with glutamate is going to generate glutamine as catalyzed by GS, or glutamine synthase. We asked whether this is indeed what's happening. Is GS active? So we labeled the cells with ammonium chloride that was heavy, so 15N ammonium chloride, and we asked whether we can trace it to glutamate and to glutamine. Although the levels of M+1, so labeled glutamate, were similar between the non-adapted and adapted clones under low-low conditions, we saw a significant difference in labeling the labeled fraction of glutamine. So you can see here about 30% versus 8% labeled glutamine, indicating that, so this M+1 is coming either from labeled glutamate with upon condensation with unlabeled ammonia, or vice versa. So labeled ammonia with unlabeled glutamate, but we can also see M+2, which is the result of having the dual labeled substrate. So both ammonia and glutamate are labeled. This leads to the generation of N+2 glutamine over here. And again, there's about 8% versus 2% labeled fraction in the adapted clones versus non-adapted clones, only under low-low conditions. So far, we have shown evidence that the adapted pancreatic cancer cells have higher fitness in vitro and in vivo. They have altered their metabolism to look more like as if they were under nutrient replete conditions. They have increased DNA synthesis. How do they do that? They use the amino acids, use the nitrogen from, for example, branched-chain amino acids to synthesize purines and pyrimidines, and also to synthesize glutamine. Because glutamine is essential for cellular growth and for nucleotide synthesis. And they also have increased mTORC1 signaling. Since we know that adapted cells have increased mTORC1 signaling and GS protein levels, these two always mirrored each other in our study. So we started thinking that perhaps mTORC1 is affecting the levels of GS. And we know that mTORC1 is a master translational regulator. First, we looked at the transcription, so mRNA levels of GS. We inhibited mTORC1 with taurin-1, and we found that the mRNA levels of GS were actually increased. But then, when we looked at the protein levels, we treated first the cells, the adapted cells, with cyclohexamide, which is a translational inhibitor. And surprisingly, there was no change. GS was stable when we treated the cell with cyclohexamide for up to 8 hours. And only when we treated the cells with taurin-1 to inhibit mTORC1, did we see a significant decrease in the levels of GS, indicating that mTORC1 is regulating GS at the post-translational level. And perhaps we wondered, is mTORC1 preventing the degradation of GS? And so if we inhibit mTORC1, then we see its degradation. So we repeated this experiment, but now, so you can see here that taurin-1 is leading to a decreased level of GS in the different clones of adapted clones. So I'm comparing these two bands and these two bands to each other. If we now add MG132 in the presence of taurin-1, MG132 is an inhibitor, is a proteasomal inhibitor. What do we see? We see that we can rescue the levels of GS by preventing proteasomal activity, by suppressing proteasomal activity, indicating that mTORC1 is indeed preventing the degradation of GS. And in fact, when we look at GS in the presence of taurin-1, and especially in the presence of taurin-1 and MG132, we're able to see that GS is polyunicotinated. So the model is that mTORC1 is preventing the degradation of GS, we inhibit mTORC1 with taurin-1, that leads to its ubiquitination and to its proteasomal degradation in the adapted clones. So because we saw that there was sustained mTORC1 signaling in the adapted clones, despite the fact that these cells were chronically deprived of glucose and glutamine, we started suspecting that we may have perhaps selected for inherent mutations that affected mTORC1 signaling. So did we select for cells that had mutations in mTORC1 signaling, allowing them to sustain this activity? So to address this question, first, what we did to have a very quick assay and address it, we generated this assay that we called adaptation reversal. So we took the adapted clones and we cultured them back. So remember, adapted have been chronically cultured for many, many passages under low conditions, low glucose, low glutamine. We put them back in high conditions for at least a minimum of 4 passages or even more. And past those 4 passages, we then treated them for 24 hours with either high or low to assess their phenotype in terms of signaling and metabolic and the protein levels. So in terms of signaling, it was evident to us that the higher the passage, so with subsequent passages in reverse adapting them in high conditions, and then we treated them with low, right? So we're looking over here. So these passages are 0, 1, 2, 3, 4, indicating how many times they were reverse adapted in high, medium, and then treated for 24 hours with low. So you can see the higher the passage, there's a gradual decrease in the mTORC1 signaling, as evidenced by a decrease in the phosphorylation of its substrate S6K and also for EPP. And as would be expected, because CAD is the substrate of S6K1, we saw a similar decrease in CAD, the rate-limiting enzyme in pyrimidine synthesis. Also similarly, mirroring mTORC1 signaling is a decrease in glutamine synthetase levels. So this assay simply indicated to us that it is most likely that the adaptation of these cells to low, medium is non-genetic. So that was super exciting to us. Importantly, wanted to see if the phenotype itself, the enhanced proliferative fitness, was indeed reversed, not just the signaling. And yes, so this is just a representation of one of the cell lines, showing that when we reverse adapted the clone, so here in brown, you can see the reverse adapted clone 5 has an intermediary phenotype between the low fitness of the non-adapted clone 2 and the higher fitness of the same clone adapted clone 5. Consistently, transcriptionally, over 1,000 genes that were the top genes that were altered upon adaptation, either by an increase or decrease in their levels. When we looked at them in the adapted versus the reverse adapted states, we could see that about half of them demonstrated, so 1, 2, and 5 clusters right here, demonstrated partial reversion of this phenotype. So there was an induction, there was partially reversed in the reverse adapted clones, as you can see over here. So this is indicating that perhaps the adaptation requires only a portion of these genes to happen. But I will get back to this in a little bit. Because now we have evidence that this is non-genetic, we confirmed it indeed through whole exome sequencing, and I'm not showing it over here. And we found that there was indeed no high impact or moderate impact mutations in the adapted clones, whether it's an mTORC1 or upon reverse adaptation. In fact, the clones cluster together according to their number and not according to their adaptation, indicating that if there were any mutations, they were minimal. They did not have a significant effect on the phenotype. And so now that we have evidence that this is non-genetic, as we know, inactive transcriptional elements reside in closed chromatin, whereas active transcription happens in open chromatin. So to assess this, we performed a genome-wide chromatin accessibility assay termed ATTAC-seq that allowed us to see the landscape of open chromatin. And we saw a dramatic change in the landscape of open chromatin when we go from non-adapted to an adapted state, as you can see here, with gains of loss of sites near thousands of enhancers. So this was exciting. But then we thought, what would happen if we were to look at the reverse adapted clones? And to our surprise, reversal at the chromatin level was infrequent. It was very rare to see that. So indicating that these changes at the chromatin level were stable and that perhaps they are maintaining an epigenetic memory. And what we think is happening is that for us to completely revert the phenotype at the proliferative fitness level, the signaling level, proliferative fitness level, transcriptional level, and epigenetically, you would need to culture these cells, reverse adapt them for many, many, many more passages in high. So it takes a much longer time. It is gradual, but it takes a much longer time to completely revert to the non-adapted phenotype. So now that we know that this phenotype is reversible, the adapted cells have enhanced fitness, and that GS is induced so that it helps the cells synthesize nucleotides, etc., and that mTORC1 is key in stabilizing GS. We wanted to know whether really GS was required for what is its exact contribution to the adaptation phenotype. So to address this, we treated the cultured cells with an inhibitor termed MSO, and we did find that inhibition of GS with the inhibitor affected the growth significantly of the adapted but not the non-adapted cells. But then we wanted to know whether this was also happening in people. So we transplanted or injected pancreatic cancer cells that were expressing hairpins or control scrambled in the pancreas, so that we can inducibly knock down GS post-transplantation. And the way we would do it is by doxycycline treatment in the water of these mice. And as you can see, so I think we treated the mice 5 days after we injected the cells. As you can see, this is the control. There's no significant difference in the size of the tumor upon, I think this was about a month after the injection of the cells. So 5 days after injection, they were treated with dox, and then another 25 days later, the tumors were harvested. You can see that with this hairpin without dox, so this is the tumor size, so this is the control. But when we treated the cells with the mice with doxycycline in their water, there was about 9-fold decrease in tumor size. Also consistently, although it's not exactly the same, and we all know that hairpins look very different, but again, we have a very similar phenotype where the tumors grew to this level, but when we treated the mice with doxycycline to knock down, silence GS, we see about a 6-fold decrease in tumor size. So indeed, GS could be a therapeutic target in pancreatic cancer. It is hitting the cells that are adapted to nutrient deprivation. And as we know, when the cells are grown in vivo, they are indeed nutrient deprived. So the adaptation in vitro is a mimic of what is happening in vivo. So the model that we came up with, so a summary of actually what I presented here, is that adaptation of pancreatic cancer cells to nutrient deprivation results in the activation of mTORC1, or they sustain the activity of mTORC1, which prevents the degradation of GS, which is required for, at least partially, for the enhanced fitness of these cells, both in vitro and in vivo. Non-adapted cells, on the other hand, do not maintain mTORC1 signaling, and with a decreased activity of mTORC1, GS is proteasomally degraded, resulting in a decreased fitness of these cells, both in vitro and in vivo. For us trying to extend these studies to human pathology, because all I've talked about is human cancer cells grown in culture, human cancer cells that were injected in animals. So we looked at mouse, basically. So now, can we extend it to human pathology? So we acquired, through a collaboration with MGH, about 127 tissue microarrays of resected tumors from pancreatic cancer patients. and we immunostain them for GS. And just to give you an idea, GS stains islet cells strongest. Acinar cells have modest staining. How about the tumors? The tumors had a wide variety, very, very heterogeneous staining, going from strong, modest to weak, with an average that was actually lower than that of the acinar cells. On average, there was no association of GS with either tumor size, involvement of lymph nodes, so invasion basically, or which can be summarized as tumor grade, nor was it associated with survival. So that might sound surprising to you, and as it was at the beginning to us, but then if you think about it, the fact that GS was heterogeneous, the staining for GS, this expression is heterogeneous in the tumor, is actually consistent with a heterogeneous or gradient of nutrients, of glutamine levels. We know that in the core of the tumors, glutamine levels is significantly lower, at least twofold lower, 2 to 5-fold lower than the periphery. And so perhaps future studies, if we weren't able to acquire whole sections, would enable us to show that this heterogeneity is the result of proximity or distance from the blood vessels, and hence access to glutamine. So you can think of the tumor as having clusters of cancer cells within the tumor mass, okay? And depends really on the location of these cancer cells, where they are, and their proximity to the blood vessels, that then they do express or they don't express GS. So you can think of them as the tumor is growing three-dimensionally, it's going to reshape the structure of the tumor. It's going to push these cancer cells away or closer, further away or closer to the blood vessels. And you can think of these clusters turning on and off slowly, not like a Christmas tree, but takes a long time as the tumor is growing, depending on when they are, how close the glutamine is to them. So we would love to look at this in the future. And we think that this is perhaps why we saw this huge, wide heterogeneity in the staining of GS, this expression, and we were not able to correlate it to patient survival or tumor size or tumor grade, etc. So despite the fact that GS cannot be used, we think, as a prognostic marker, it is still indeed a therapeutic target. Why? Because even though it does not tell you whether this is more aggressive cancer, etc., because of its heterogeneity, still when you inhibit it, you are going to target these most aggressive pancreatic cancer cell clusters in the tumor, because these are the ones that are the most adapted and have survived harsh nutrient deprivation. So again, even though it's not a prognostic marker, we could think of it as a therapeutic target in PDAC. And so with that, I want to end with this model where we think that adapted pancreatic cancer cells acquire epigenetic alterations. So epigenetics is here first. Upon it, and it sets this adaptability switch, or actually a dimmer. And it endows the cancer cells, gives them this metabolic flexibility to nutrient-deprived conditions. So where here they turn on mTORC1, GS, higher fitness, or to now switch back to nutrient-replete conditions slowly again, because if you remember, we have this epigenetic memory, or it's the chromatin changes are actually stable, but through consecutive or progressive de-adaptation with increased levels of glutamine, glucose available, then they revert back to this non-adapted state so that they don't waste much energy, right? And then because they have this metabolic flexibility, they have this higher fitness. And we think that that is perhaps one of the reasons why pancreatic cancer is so aggressive. Big questions for the future we are thinking of is, what is it that happens epigenetically? Is there a leak in a transcription factor that allows these adapted cells to survive and then drive this whole metabolic program that gives them this metabolic flexibility and enhanced fitness? Other smaller questions are, what other factors are involved other than GS? We know that GS is not the answer because this is much upstream than GS and it's one of the contributors to this fitness. What other factors are involved other than GS, or how does entropon actually regulate GS at the proteasomal level, etc. So with this, I would like to end and acknowledge everyone that has worked hard on this project, particularly the Kalaany lab members, Min-Sik Lee and Pei-Yun Tsai, who headed this project, and with the help of other lab members, Insian Akli, Ashley Adler, Pete Del Nero, our small animal imaging core at Boston Children's, our collaborators at MGH, at Dana-Farber, at the School of Public Health, the Harvard School of Public Health, the Broad Institute, and the Whitehead Institute, and importantly, our sources of funding. And thank you very much for your attention, and I look forward to your input, feedback, and questions through Twitter.